technical questions. So as I say, welcome to the ZP Developer Zone. We like to do this every week, and I'll sort of essentially just jump straight into it. So some of the questions that have come in this week are, I want people to remember that we do have our ZP Academy. There's a couple of free courses up there, especially if you've got questions around sort of electrochemical techniques. We have our webinars, so we provide this as a kind of we try to provide this as a sort of service to kind of answer some of the lighter questions that come in. I realize sometimes the questions actually go quite, I want to say almost quite deep quite quickly. So then that requires things like the workshops. We do have collaborations. Now collaborations are much more with universities. So for example, in Europe, there's these Horizon 2020 projects. In the UK, for example, we sponsor sort of PhDs, so we do have a sort of academic friendly policy. It does help, obviously, when there's government funding, in place, and that sometimes makes it easier in certain parts of the world than other parts of the world. We do offer jobs through our really through LinkedIn, I would say, is where the place we advertise that. We have our ZP Developer Zone, and we have our workshops. I'll go a little bit quicker today because there's something like 9 questions. I do want to highlight the workshops, and I also want to highlight that if you see a link today, I just want to say hi to Hitcham. Good morning, Hitcham. If you see a link, it will be already underneath the video. So workshops are great because that's where we can sort of dive into things a lot deeper. We do have a workshop on the 8th and 9th of February. And in that workshop, we will be covering things like smart wound care and the measurement of pH, for example, in bandages. I just want to say also a good morning to Aftab as well. Thank you, Aftab. Nice to see you this morning. And I specifically want to say to Aftab that we're going to be covering some questions today around capsaicin and the measurement of chili. But anyway, question number 1, somebody wants to measure pH, temperature, and conductivity in bandages. So ZP adds most value when it comes to conductivity and pH. Anyone can monitor temperature. So the value we add really is around the pH and conductivity. We did have a questioner who wants to measure calcium, ORP, oxidation reduction potential, NO, nitric oxide, and hydrogen peroxide in the plasma phase. And I will talk about, a lot of what we do is actually, we've worked in the liquid phase. That's where I would say 95% of our experience is. The gas phase, the vapor phase, and we use the plasma phase, but we don't often measure in it. So that one, I just want to say hi to Patrick this morning. Thank you, Patrick. I think some of your questions are tied up in here as well, Patrick. Question number 3 is detecting the survivability of cells. So somebody wants to essentially go through a sterilization process and understand whether cells survive that process or not. I want to touch upon entrepreneurialism. And I'll sort of, I'll touch upon, that. Also, training support and screen printed electrodes. I will touch upon the quality of screen printed electrodes. And I'll also just very quickly go through a list of biosensors that somebody's interested in. Measurement of pH in flow. This is something we'll sort of answer a little bit more deeply. And then I say question number 8 here is precision, accuracy, and error in capsaicin measurements. So I will dig into that a little specifically for AFTAB, and then problems with glucose measurements in SMBG. So somebody's wanting to know what are the material science problems in self-monitoring blood glucose strips, and I will touch upon that this morning as well. First of all, somebody wants to know about how to measure pH in wound care. They say here conductivity as well, but I thought, well, focus on pH. Conductivity is a sort of, they're both pH and conductivity measurements are straightforward. The only reason I'm skipping over temperature a little bit is just, most of a lot of our tech has a temperature sensor sort of built in anyway, so that's essentially for free temperature sensing. When it comes to wound care, we've talked about all sorts of biomarkers in the past: oxygen, lactic acid, uric acid, pH, purine, conductivity. These are all sort of useful biomarkers around the healthiness of whether a wound is healing or not. So I won't go over it too much, but we have taken a critical look at our pH sensors and said, will they actually work in a range that's a kind of indicative of the healthcare of a wound, where a wound is at like pH 5, it's kind of considered healthy, and if it's kind of more alkali, i.e., has a higher pH than 7, then it's considered to have a sort of problem. And there's plenty of, let's say, difference between those pHs that we could get a fairly accurate clinical device out of this. We have the kind of components, we have the pH sensors, we have the microfluidics if it needs to be adhered. We do have electronics, easy flex, and then the single purpose circuit board here. So there's really no issues with it. I think for years people have been talking about doing smart bandages, smart plasters, smart wound care, and I do see a change now in the market that people are generally trying to work upon this stuff. So we have all the components. It's just when you try to get into a medical device, it's not really enough to kind of just buy this stuff and integrate it. You have to work with companies like ZP, especially as we are ISO 13485. And we are able to put all this technology together into wearable sensors, apps talking to Julie sending the data to the cloud, doing data science on those apps, and essentially extracting useful information out of it. For example, whether that wound is infected or not. So we do have that stack of technologies. The pH sensor, for example, is an open circuit potential measurement. So I put a link here. There's a video here that shows us doing a lab experiment around that. But we do cover pH sensing in our workshops, and that's an example of a potentiometric measurement, specifically open circuit potential, so it suggests it's high impedance, and that is covered in our workshops. When it comes to sort of wearables, people are very interested in whether these things are flexible, so they can kind of conform to the skin. So it's really of no issue for us to be able to make at least the conductivity and the pH sensor in a wearable, flexible format. This is exactly what we expect. But as I say, it does only really work if people engage with us properly, I would say, on projects. Question number 2 is kind of interesting because they want to measure calcium, ORP, oxidation reproduction potential, nitric oxide, and hydroperoxide. First of all, on wire sensors, we have these. Let's say this is one of our wire sensors. I mean, they're constructed by literally having a wire, having an insulator, and having what's called a sort of a reference electrode around them. Now, I don't show it. Now, what we do is we actually functionalize the surface of what's called the working electrode to make it specific to the analyte of interest. But what I'm trying to show here is actually when we're using these wire sensors, they're often in a liquid, and that liquid is often obviously kind of blood, buffer, urine, teardrop, interstitial fluids, but they're in a continuous liquid phase. So I want to sort of touch upon the idea of plasma, because I would almost describe it as a sort of, I don't want to say, it's almost like a discontinuous phase. There's definitely spaces between the ions. If we were talking about our screen printed electrodes, then we'll take our sort of screen printed electrode, we'll often functionalize them. This is an experiment that we do actually on these workshops, where we would take these screen printed electrodes that have been functionalized, for example, calcium. We'll put it into a beaker, and I might change the example to glucose in this case, because the data I'm going to show is glucose, but we'll have it in a liquid phase. And we'll do a series of additions where we're adding more glucose and the signal kind of goes up. So I've shown the wire sensor, and I've just emphasized the fact we're in a liquid, and now I'm emphasizing our screen printer electrodes and saying they're in a liquid. Now I want to touch upon plasma. If you're going to do plasma, if we're doing gas, now gas is not a plasma, but if we're doing a gas electrode, what we do is we put a solid electrolyte onto it, because the liquid is essentially our electrolyte. But in a gas phase, we don't have a kind of continuous liquid. We essentially have gas molecules that are separated from one another. In between them, I almost want to say this, I don't want to say vacuum, but there's not molecules between there. There's not this ability for ions to sort of have a continual electrical connection between our working electrode and reference. So if we're making a gas sensor, we actually often put a something like a sort of solid electrolyte on top of there. So when you look at plasma, for example, sort of just done a little cartoon here, obviously, of positive and negative cations and anions. There's this, I've shown it in my simplistic way of actually there's, even though the plasma is on this electrode in this case, it's not continuous. We don't have a continuous phase. In order to detect a plasma, we'd have to actually have a solid electrolyte on there. And then the plasma would essentially come into this kind of electrolyte, and then potentially we could detect it that way. That all said, we have done solid electrolytes for gas phase molecules. So something that's quite similar to nitric oxide is carbon monoxide. And so this is, and I know I've changed courses here. If you want to do gas phase work, then we'll put a solid electrolyte on top of the sensor. And then we've tested one here in the gas phase where this is the gas. We turn the gas on, we get a signal change, we turn the gas off, the signal essentially changes, but it does require a solid electrolyte. I have put a link to it there. So all that said is that calcium, ORP, nitric oxide, and hydroperoxide, we have not measured these things in the plasma phase. And the reason I bring that up then is because I don't want to disappoint, essentially. Plasma is quite energetic, so I'm not entirely sure what it's going to do to our sensors. We do use plasma. We use plasma deposition sometimes in the functionalization of sensors. We obviously use sort of plasma to treat surfaces, as you would already know, to make them more hydrophilic. We do use plasma, but the idea of measuring these molecules in the plasma phase, or these, I say, properties, especially the ORP, we've not done it. I just want to sort of slightly just show you something that's kind of related, but not the same as. I've so far said that most of what we do is in the liquid phase. I have touched upon the gas phase. I've kind of said we haven't had experience of doing in the plasma phase. It doesn't mean that people shouldn't try it. We just, we talk about experience. But I just wanted to highlight something that was kind of interesting a little bit, that we are detecting molecules in the vapor phase. Vapor phase, as you know, is essentially sort of liquid droplets sort of carried in the gas phase. So we do detect in the vapor phase. What we're particularly doing in this application is actually condensing the vapor back to liquid and then detecting it, and that's something that we've done. And that product, for example, is on the medical market. That people can use it for the detection of COPD. Just as a side note, we're actually detecting hydrogen peroxide in that case as a biomarker for COPD, chronic obstructive pulmonary disease. I do want to touch on that we do have the calcium sensors, but that we're only using them in the liquid phase. We have the ORP sensors, but we use them in the liquid phase. This is a slightly different question. The question I would like to know, can we use techniques like EIS, electrochemical impedance spectroscopy, to check for pathogen inactivation, which they've done by a sort of an energetic sterilization? So let me sort of touch upon that a little bit here. That it's worth saying that EIS, electrochemical impedance spectroscopy, is used to measure cells and also the viability of cells. This is I've got to say immediately that these images are from a company called Aber Instruments. I don't have an association with Aber Instruments, but I just wanted to say that the principle in fact is used. They use it for monitoring cell culture. I'm sure they do, and I think they do a very good job of it. I think they're distributed through Sartorius, from reading the literature. Anyway, the idea of using EIS or impedance spectroscopy for measuring cell count and cell viability is a thing that's done. It's something that we have looked at in the past, and essentially I think it works fine. That you can take many of the kinds of electrodes that we have. You can put a sort of an EIS signal between the electrodes. If the cells are intact, and they being intact is important if they're viable is maybe a stronger word for it. If the cells are viable, then we should be able to detect and also not only know that they're viable, but also do a cell count on it. But you do have to essentially sort of calibrate it and um, optimize it in the application of interest. It's not enough just to kind of start shooting at this. You have to sort of obviously do some science around this. Um, but in principle, it can be done. I think we've got lots of technologies that would allow this to happen. Um, we do have a sort of Sense It All platform that would allow you to take a sample, um, check the pathogen, potentially check the pathogen load first, and then actually do the sterilization and check the pathogen viability afterwards as well. So the technology stack exists, but what we find is we have to kind of tune these things up for the particular application of interest. Question number 4 was really about entrepreneurialism. This was a tough question, really. A nice guy that has done his PhD in Cameroon, is kind of interested in starting a business, was asking whether to do it in Cameroon, whether not to do it in Cameroon, and also sort of being very modest and sort of saying, well, I've only got a background in like electroanalysis, so not necessarily in making sensors. I wanted to put a picture up of Elon Musk, but I really, I don't know if I've got any pictures of Elon Musk that I can put in the public domain, so I have to put a picture of myself up. It's just a really, these are the sort of 7 habits of highly successful people. I'm not suggesting I'm at all highly successful. I just want to say that, when people are sort of umming and ahhing and worrying about things a little bit, when I look at this list, I mean, it says sort of be proactive. And I generally think that this guy, for example, has been proactive by reaching out to me and asking me, some questions. So that's a good sign. And I think it's begin with the end in mind. That when what that's quite good is because starting a business, you have to have a kind of why am I doing this? Where is this actually ultimately going? Um, so that you can always be steering yourself, because life can obviously take you in many directions. But if you have a sort of a view on where you want to go, then it can help you sort of, even if you sort of feel like you're tilting off slightly, you can sort of tilt back again or steer back again. So, be proactive. I think this gentleman's already described that. If this gentleman has the idea that actually he wants to make sensors, then he's begun with the idea in mind, or the end in mind, I would sort of say. When they say put first things first, I do feel like, if I looked at Zimmer and Peacock, we've started off very modestly. Um, and it's grown and it's grown and it's grown and it's grown and it's grown. So you do have to just essentially start. Um, Now that's that starting point. That's the problem. Nobody can really teach you that. You have to sort of do it. Let's say. I mean, think win-win is just a situation really where you do have to have a mindset that if you're going to be successful, then you have to bring other people with you. There's just no doubt about it. It's really your network and the people around you that will make you successful. You won't be successful in a vacuum. Um, Seeking to understand, I mean, it's just as important to listen as it is to speak. And I was told once that, we have 2 ears and 1 mouth, and I should use them in that ratio. So I do understand what that means which is, you know, when you are trying to do something, the more you listen, then the more you learn. Um, and don't always try to sort of dominate every conversation. I'm sure this gentleman doesn't, but it's just one of those lists of sort of height of successful people. Synergize, I think, is really linked to win-win. You need to work um, in partnership with other people. So we have a very successful partnership, I would say, with Technando, and it's a synergy because we contribute, they contribute, and the sum of 2 things is greater than what either one of us acting alone. So synergy is important. When it says sharpen the saw, they do say about continuous self-improvement. Um, I mean, I know that they say, a lot of, billionaires read, a book a week. For me, I think self-improvement is bigger than that. When new technologies pop up, as I'm sure many of you do, chat GPTs, like an example, it's just immersing yourself and always trying to stay current and trying to stay, up to date all the time. So I don't, nobody has a formula for success in life, but I would say one thing: it is if you look at Elon Musk, for example, he's from South Africa and he's successful in the USA. Sometimes if you want to grow a business fairly rapidly, you probably have to try and get yourself into some of these major economies. Elon didn't grow Tesla or PayPal in South Africa. I'm sure he still does lots of work there, but he grew it in the US, and the US was the biggest market in the world. It is easier to be successful in business the bigger the market you're in, or the faster your market is growing. So India, for example, it is actually a big market and it's also growing. Um, so I would say it would sometimes, if you feel like your market is stagnant and even shrinking, that is a hard place from which to then grow a business. So it's just the reality of it. And Elon Musk, I could use as an example. That he's South African, but he grows his businesses primarily in the US. Question number 5. Um, Somebody really wanted to know about getting trained up and also how to get high quality electrodes. I'm super biased. We do have some resources around screen printed electrodes. If you see a link in this webinar, I will also put that link underneath the videos as well. So we do have a couple of links, resources there. At ZP, we do make a really good, for example, hyper value electrode. And again, if you see a link, it's underneath the video as well. I mean, what's happening with us is, we have a general sort of manufacturing procedure. We do OQC, outgoing quality control, and it's not, and we also have a bespoke system called Julie, so we're analyzing our data as well. And then we're sort of, packaging and shipping it. Many people, the problem with screen printed electrodes is many people can do what's called visual inspection, where you can actually, look at the electrodes, and if you see something that's blatantly wrong, you don't have to ship it. But at ZP, we do a little bit more. We actually do a lot of electrochemically testing these electrodes as well to make sure that they're fit for purpose and that we can actually ship them. We do all the statistical analysis on them, and we will reject, we will accept. So there's a lot of effort goes into things like our hyper value electrode. I've said it so many times, but the biggest consumer of screen printed electrodes at ZP is ZP, and that's why the quality is high, because we care about them. If people are if we're turning them into biosensors, we'll also functionally test those biosensors as well. So, for example, glucose testing here. And it's probably worth saying, because we are electrochemists, we end up testing them in many different ways. So we do something called wafer mapping, which is, we'll make a batch of electrodes, we'll test the cost of the batch, make sure that everything is good. We'll do what's called heat mapping, so we can see if there's any sort of problem spots. So we've really characterized these electrodes before they've even shipped to our clients. So for example, if we didn't do this, then our chili sensing wouldn't be quite as accurate and precise as it really is. And it's probably worth saying that if you are looking for training around screen printed electrodes, it's not something you can just talk about online. You have to kind of come along to a workshop and get trained up. Question number 6, we did have a list of somebody who was interested in electronics. So we've talked about the switchable or the single purpose electronics. They want to know about glucose sensing, links to glucose sensing. They wanted to know about ATP, so adenosine triphosphate, NADH, nicotinamides, pH, phosphate, redox, oxygen lactate. And if you're looking for generic sensors as well, we also sort of gave them a direction towards general screen printed electrodes. I mean, if you're in academia, most of the time people want to invent. So really they end up building sensors themselves. We did get a question about measuring small volumes. It's probably worth saying that when we're testing things like pH sensors, we're often putting on something like 30 to 50 microliters, because that's a sort of typical drop size. We'll put a drop on there and we'll get a signal. So we don't have any problems with flowing. With the volume size. We can, for example, we're making sensors today for clients where the volume under test is 4 microliters or 1 microliter. We're obviously doing this in microfluidic devices, but no real sort of issue, let's say. Our sensor, especially the pH sensor, will withstand flow cells. There's some quite nice videos of Andre here assembling a flow cell. He makes an electrochemical measurement in it. I mean, it's quite a nice video in some ways, because much of what he's doing here is, it's very open as to what he's actually doing. So the flow cell, putting sensors and pH sensors in flow systems is really of no, I would say, big issue. It's probably worth saying, though, that if you're really a corporation trying to do product development, it's never really works to just buy some stuff off the website and sort of put it together yourself. There will have to be some NRE activity with ZP. We almost describe it as product as a service, that we do have all these products, but bringing them together and making them work in a particular application, that can be something that you have to sort of engage with us on. Right, measuring the hotness of chilies. It's kind of interesting because at ZP, we have a big background in medical diagnostics. We are what's called ISO 13485, so we're certified for the development and manufacture of medical diagnostics, which is really important because it puts us at a really quite a high level in terms of them working in other spaces. So if somebody is trying to measure the hotness of chilies, there's sort of three techniques. Analytical, using HPLC, high pressure liquid chromatography. There's panel testing, where people are literally tasting this stuff and scoring it, and then the food sense, which is something that we manufacture. As I say, we're actually, when you look at the food sense technology, you can see that it's actually got some strong similarities to glucose sensors for diabetics, and that's not by accident. That's a sort of reflection of our POCT, our point of care test background, or rapid diagnostic testing as well. So we obviously make most of our money in the medical space, and then we bring that kind of technology across to the food industry. So when in developing this technique, one of the things we have done is we own an HPLC system, a high pressure liquid chromatography. We've taken capsaicin. You can buy capsaicin from Sigma Aldrich, made up standard solutions, so we test those. We've also tested or extracted, pastes and chilies and powders and sauces, and just check that we correlate with HPLC, high pressure liquid chromatography. So that was part of our sort of validation to make sure that our methods were also telling the same story as the HPLC. I just want to touch on accuracy, precision, what people expect from an analytical technique is, that you have good accuracy and good precision. So by accuracy, it's telling you the truth. And if you did multiple tests on it, they would all tell you a similar result. So that would describe as accurate and precise. You could actually have a technique that was accurate, but imprecise. So you made 3 measurements. So for example, I made a measurement that was 4, 5, and 6. So maybe I'll do it in Scoville heat units. 4,000 Scoville heat units, 5,000 Scoville heat units, and 6,000 Scoville heat units. The average of those 3 is 5,000. If the truth was 5,000, then you would describe that technique as accurate, but actually you would also describe it as imprecise, because you had to do 3 measurements in order to kind of average out all the errors and get an accurate result. So you would describe that as accurate and imprecise. You can have inaccurate but precise. So for example, you really think that the technique, let's say you think that the result is 64,000 Scoville heat units. You make 3 measurements and they all say 92,000. You would describe that as it's precise because actually the technique was consistent, but it's inaccurate because it wasn't giving you the complete truth. And then you would have inaccurate and imprecise, where it's neither telling you the truth and neither is it repeatable either. And that's a kind of worst case scenario. Now, I think HPLC in the food industry, especially if you're not doing the HPLC yourself, especially if you're sending it out to labs, we have noticed this. When you send raw materials to a lab and you get a certificate of analysis, if you send it to different labs, you can actually get different results. It's no insult to the labs. It's hard because actually they have a chili powder, a chili itself. These are very heterogeneous samples. So you think you're sending the same sample to the same lab, to different labs, but in fact it's heterogeneous. So I want to defend these labs, but I would say that if you sent samples from the same batch to different labs, you might actually find out that they're coming back with different results. So you've got to be very careful with certificates of analysis from things like HPLC labs, especially on raw food ingredients. It's a different thing with things like pharmaceuticals, where they're highly pure, highly homogeneous samples. But it's much harder with these sort of impure, heterogeneous samples. So I'm saying here, like, even if you're doing a certificate of analysis and trying to work out the Scoville heat unit from a lab, if you try 3 labs, you actually might get 3 different results. So what does that say about the accuracy of the results that you're actually getting? There's also another challenge that one can do is actually send the same samples from the same batch to the same lab, but don't, you almost have to blind the lab. The lab shouldn't know this. If you send it, and if you send it over time, that can also help. And you'll actually find, when I say send it over time, I literally mean that. So if you send them all at the same time, they'll all go in the same machine the same day, and you'll get fairly consistent results. But if you send them 1 or 2 weeks apart, then you'll actually find that there's more variability because of the separation in time. What I'm saying is, even if you go to the same lab and you'll think you're sending them the same sample, especially if you send that same sample separated in time, and the lab doesn't realize it's the same sample, you'll actually get 3 different results back. Again, it's no insult to the labs. It's just the method of HPLC analysis. There's a lot of extraction time. There's a lot of column. They have to calibrate that column. They have to inject it onto the column. They have to interpret the signal. There's quite a lot of steps involved in this. And there's much more sort of drift or inaccuracy in those results than people really believe. So what's the implication then? So the implication for us at ZP is, that somebody can have a certificate of analysis, and they can essentially know, they can think they know what the Scoville heat unit is. So I put like 64,000, and it's measured on HPLC. Now you come along with the food sense, and you get potentially a different value. For example, in here I've put like the idea that they got 98,000 Scoville heat units. So you're like, well, who's correct here? Well, the thing is, in food testing, it's more important to probably be precise, rather than accuracy. What I mean by that is this: that if you've been using HPLC for a long time, you have a timeline and you have Scoville heat units, and you put a sample in, it gets tested, you have a Scoville heat unit. You put another sample in, and get the same, similar result. You get the same result. So everyone, at that point, they're happy. Now you bring the food sense along, and actually you again get precision because you get the same result. But whereas this was reading 64,000 and this is reading 98,000. Now the question is who is accurate? What's more important is actually, if you're trying to use it for quality control, you probably want to just be consistent. That I've measured these samples. I want my method of analysis to be consistent between my 2 methods, but there's actually an offset between here. Now this offset exists because people would think that HPLC was really like a gold standard. I would say, if you think it's a gold standard, try challenging the analytical lab by same sample, send over several weeks, and see what kind of results you get back, and you'll probably find that the results are variable. But the question is, then you bring a new test in, which is much lower cost, much quicker to do. You get an immediate result, but it's offset. What do you do in that situation? That's why we brought in the response factor. So if somebody wants to be consistent with their old results and their new results are again consistent, but there's an offset between the 2 results, what you can then do is apply what's called a response factor. And the response factor then will say, okay, take this result, times it by 0.65, which will move it in line with the original results. Don't like to do that because actually I think the food sense is accurate. But when people have historical data, they want to be in line with the historical data, and so there, you can do this this correction. What it basically says is that this method is more sensitive than this method, which is not unusual, I think, and therefore we have to almost tune down the sensitivity so that we get in line with the HPLC method. So that's one way of sorting out an offset between HPLC and, for example, food sense. But don't think the HPLC system is just because you got that certificate of analysis. What's interesting is certificates of analysis, they don't always show you their error, because when somebody says your Scoville heat unit is 64,000, plus or minus what. And now they might, and then you've got to prove that that error that they're telling you is actually true. So be careful. I also want to talk about relative error. So let's say you had an expected Scoville heat unit of 110,000 Scoville heat units. You measure at 97,000 Scoville heat units. The relative error between those is, this is the expected result. You subtract your measure result. You divide that by your expected result, and you actually get a result of 0.13 or 13%. That's probably a very acceptable relative error, I would say. So numbers, numbers don't have to be exactly the same. You just need to look at the relative error and sort of think, is this acceptable or not? And when you've got a heterogeneous sample like a chili, it's not unreasonable to have differences like that. Especially when that's measured by HPLC and that's measured by food sensor, do different technologies. You could use the correction that I showed you on the earlier page and actually put a sort of sensitivity factor in of something like 1.1 or 1.2, and that would actually raise it, but I don't think you need to. I think you can live with 13%. Question number 10 is about materials issues with glucose strips. So at ZP, we work on two types of technologies: CGM, continuous glucose monitoring, or continuous glucose monitoring, and glucose strips. And the question is what materials science problems are there? So I did do a search on this, and the problems with SMBGs or glucometers is the test strip is damaged. The test strip is damaged or used. The test strip is expired. The test strip is not inserted into the meter correctly. The test strip is moved, so shaking during usage. The test strip has not been kept to the right temperature. Not sufficient blood is applied, even though these test strips only need about 300 nanoliters of blood, which is much less than a volume. The sample site is contaminated, so if you peel a banana and then make a glucose measurement on your finger, any kind of glucose that came off that banana skin will also confound the sensor. Low battery, that's just to sort of change the battery problem. Alternative sites, these glucose strips are calibrated to work on finger prick. So if you take the blood from somewhere else in the body, you'll get an inaccuracy, and then just sort of squeezing too much on the finger so you can't get the blood. The reason I list that out is because the inquirer is looking for a material science problem with glucometers or glucose strips. And the answer is that these technologies have been around for like so long, almost 40 years now, that all the problems are kind of gone. I'd almost say that in academia, working on glucose strips is probably not so intellectually interesting. I think it actually, so something that was on the radio this morning, there's a sort of global pandemic in avian flu. They're really worried that avian flu is going to kind of come over to humans. I actually say these are, the diagnosis of avian flu, or, the diagnosis of pathogens. These are really the academic challenges that one should be working upon. And, in the glucose strip market, everyone wants lower cost. They do. But these are not real value adds from academia. So I wouldn't actually say that, there's necessarily much merit to be made in trying to solve problems with glucose strips, because I don't really see that there's many problems with glucose strips. So question number 1, yes, we can measure pH in wounds, no problems. Question number 2, we can't promise that we can measure these analytes in plasmas. Question number 3, survivability of cells using excitation, radiation. Yeah, no problems. I think it can happen. It will work. Entrepreneurialism. It's a really tricky one, but actually I think it is quite hard to start and grow businesses, being in economies which are intrinsically quite large, Germany, Japan, USA, many countries in Europe, this is easier. And you should, and I do believe in doing good for humanity, but if you want to grow a business, then growing it in an economy that is large and there's a market exists, that is easier than trying to do it in smaller economies. We do provide training. There's all these workshops. Screen printed electrodes from us. The hypervalue electrode is a really good electrode. The Biosensor starter kit, measurement of pH and flow. I think this is just a no-brainer, really. But if you're going to do it, there will be you will have to incur some NRE charges from a company like Zimmer and Peacock so that we can support. Yeah, precision, accuracy, and error. HPLC certificates of analysis, they're interesting and they're useful and they're indicative, but they're actually not necessarily as accurate or even precise as you would like to believe. And if there's an offset between, for example, the food sensor and an HPLC, you can adjust for it. In the food industry, quality control is really more about reproducibility. Um, if the HPLC is telling you that the thing is 63,000, we're telling you it's 97,000, put in a response factor of 0.65 and they'll line up. I just don't believe they're necessarily accurate, but you need to show consistency, so that would be one way of solving it. What are the material problems in glucometers? I actually think that the real science to be done in diagnostics is in things like pathogens, and it's not in glucose. That's my opinion. So I just want to say thank you. I apologize for going on for so long this morning. But if you have any questions of ZP, you know to just reach out to us and we'll try and give it a good shot. So I want to say thank you to Hitcham, Aftab, and Patrick this morning. I appreciate your attention, gentlemen, and speak to some of you soon. Okay, take care. Bye-bye.